Good morning, everyone. I'm back again. This is Science Talk, as you can see from the title. If you're new to my channel, welcome. I do a whole array of different articles, and we have live stream discussions. Go check those out. Don't forget to hit that subscribe button. Don't forget to hit the notification bell so you'll be aware of when I'm posting new videos or going live. I really appreciate my subscribers. A lot of the articles and information that I get to do videos about come from my subscribers emailing me. If you would like to send me information and you want to stay anonymous, please let me know. If you don't let me know that you want to stay anonymous, I will mention your screen name in the videos that I do from your emails and your messages. So I appreciate you for that. My email is NicoleAli864@yahoo.com. That's NicoleAli864@yahoo.com. So let's get into this article. It's coming from Gizmodo. If you don't know about Gizmodo.com, it's pretty cool. They have a whole lot of things they talk about. It's kind of nerdy stuff, but they do have links to other articles in their network that I do not consume, do not patronize. But Gizmodo is where they discuss technology and science and STEM in a way that nerds can appreciate, I guess. So this article says this gene editing breakthrough could provide hyper-specific cancer diagnosis, and it's filed to a CRISPR file. It was published yesterday, April 19th, 2018. This is a picture of Nicholas Petrelli, director of the Gene Editing Institute and a study author. So he is in a laboratory, and I'm very familiar with that particular environment. And they go to talk about CRISPR. So if you don't know what CRISPR-Cas9 is, it's a mechanism used in gene therapy and genetic engineering laboratories to change the function and sequence structure of genes. And you can do this for any gene within the human genome, within any genome really. But specifically, this article focuses on the human genome and how to disrupt cancer genes. So genes that are involved in helping cancer cells to grow, they disrupt their function so they cannot function properly. And genes that will be decreased when cancer progresses, those genes are enhanced so that they can prevent the cancer cells from growing. They start off here saying, ever since researchers first discovered that bacterial immune systems could be hijacked to selectively change DNA in living creatures, CRISPR gene editing technology has been limited by the boundaries of the cell wall. So bacterial cells have a cell wall, and those are prokaryotes, and human cells, for instance, are eukaryotic, and we have a plasma membrane or a cell membrane. CRISPR allows scientists to cut and paste little bits of DNA, swapping out even single letters of DNA to correct disease, causing genetic mutations. So you have, they're saying single letters. So the letters that represent the DNA structure, the DNA sequence, are adenine, guanine, cytosine, and thymine, A, C, G, and T. And you have different combinations of those letters that represent genes. So if you want to Google a gene profile, for instance, you can do that, and you'll see that the gene is represented by ATGT, AGCT, AGCT, TGGC, ACC, AAG, TT, for instance. That's the representation of a gene. And those genes have different sequences. So for instance, if you had a gene that was represented AACT and you switched it to, if it was AACT and you switched it to ACCT, that gene has a new function. That's just an example. So they're saying, at least until now, all of that cutting and pasting has gone on inside the cells. So cells are able to do this cutting and pasting, repairing, and restructuring on its own. This is a natural form of selection, adaptation, and it promotes diversity among genes, therefore promoting diversity among organisms. Without that form of natural selection and alternative splicing, you would not have the diversity you see in organisms around the world on this planet. They published a study Thursday in the CRISPR journal, which shows how scientists at Christiana Care Health Systems Gene Editing Institute in Delaware released CRISPR from behind the barricade of the cell wall. They were able to quickly make multiple changes to genetic code by extracting DNA from human cells and putting it into a test tube, where a protein called Cpf1 cut into the DNA and cleared the way for CRISPR to make edits. So Cpf1 is basically an exonuclease gene. It cuts the DNA and allows new things to be introduced into the DNA. But why would you want to do such a thing? Previous CRISPR tools limit gene editing to short snippets of DNA within one single gene. Extracting the DNA from the cell could allow for more edits at one time. In the more immediate term, the study authors said this could have value in diagnosing cancer, replicating exact genetic mutations found in the tumors of individual cancer patients, and identifying exactly what kind of cancer a patient has, in order to develop a targeted treatment. The researchers are already working on commercializing such a diagnostic tool. More significantly, though, the new technique could also pave the way for new gene editing technologies that allow for removing and replacing entire faulty genes. So the faulty genes would be those that promote cancer. And they would do this to replace entire genes instead of just a snippet of DNA. That could greatly expand the usefulness of CRISPR. At the moment, while the technology has shown great promise to cure diseases like sickle cell anemia, which is caused by a single letter mutation, so one of those letters, A, G, C, and T, one of those is mutated. Like I just said earlier, if you have AACT and you say ACCT, if normally it's supposed to be AACT, but a person with sickle cell has ACCT, that second letter is the mutation. And so now you have sickle cell anemia. And they can do that with that single letter mutation, or they can go to more complex diseases, and they seem to be out of reach. So this new technology could eventually change that. But the breakthrough also speaks to the breadth of CRISPR technologies that have arrived in labs over the past year. The new tool relies on an enzyme known as Cpf1 rather than Cas9. So Cas9 was the original CRISPR enzyme that was used to cut genes, and they are deleted, and it's a permanent deletion of genes. You can also delete genes and put in a new gene at the same time. So you can delete a gene or a portion of a gene and insert something else in the place where that gene was cut out. And that happens simultaneously. So now you have a new function for a gene, or you have a new piece of gene put in that stops the production of the original gene. And then sometimes you have the cell will die, or the cell can no longer function a certain way. They say the enzyme typically paired with the CRISPR system to cut up DNA. So that was Cas9. Now they have Cpf1. It's a new enzyme. Discoveries of new CRISPR enzymes have helped to create a litany of new potential uses for the technology. For example, while Cas9 results in blunt ends which slices through the DNA like a knife slicing through a piece of paper, Cpf1 creates sticky ends that make it better suited for the removing of large chunks of genetic code. So instead of cutting through a piece of paper with a knife, you are cutting a piece of paper with scissors, but you're not cutting it straight. You're cutting it all jagged. So now you have sticky ends, and the DNA can be removed better. When you have big pieces of DNA, large pieces of a gene or a whole gene, if you have sticky ends, it's easier for the new DNA to put in or for replication to occur. It happens better that way. The ability to edit outside of the cell, researchers said, could also reveal more about the mystery of how CRISPR actually works to modify the genome. Recently, working with different enzymes has led to breakthroughs, including editing the epigenome instead of making changes to DNA itself, and using CRISPR to diagnose disease. Such studies point to the power of potential CRISPR, not just as a tool for altering the genome, but as a multifunctional powerhouse with uses we haven't yet imagined. So they have other stories here below where they talk about how editing RNA, not DNA, could cure disease in the future, and that's very important. As you know, you have 3 different main types of genetic code. You have DNA, RNA, and protein, and focusing on the DNA is not always the best way to do this editing technology, which is, of course, at a layman's term, is genetic engineering. You're engineering how genes work. You're changing how they work, or you're completely removing the gene completely. So if you edit at the RNA level, you probably have better success with gene editing. I hope this wasn't too broad, but if it was, please let me know in the comment section. I would love to have further dialogue and discussion about this. I am, I guess, a self-proclaimed nerd. I do like science and technology. I would hope that maybe this would interest you. Maybe science or this technology interests someone out there that may want to know more about it, but just may feel intimidated by the subject matter. It is very dense. It is very, very specific at certain points, but don't let the large words intimidate you. I present these articles as a way to introduce some and, just present to others science and how, these advances are becoming closer to, maybe they're coming closer to somebody because these advances are for cancer and many other diseases. There's no one really that at this time who does not have some type of cancer, some type of disease that affects them at the genetic level, at the physiological level, as far as nutrition, as far as metabolism, and so forth. So I hope that this was helpful to maybe give you interest into more science, and I'll leave it at that. I'll leave this link in the description box and talk to you soon. As always, have a great day. Happy Friday.